Where do new species come from? Well, think back to last week and consider how a population's gene pool can change over time. New alleles are created by mutation and then fixed or lost by natural selection and genetic drift. Now, imagine that one species gets split into two populations, let's say by migration perhaps. Now, because they are isolated from each other, these two populations no longer can interbreed, but new alleles continue to arise and get fixed or lost. And these alleles are different in each of these populations. This process can be particularly rapid if different selective pressures are present in these different populations. As these populations continue to diverge, they eventually become so different that they can no longer interbreed, even if they were to physically encounter each other again. And thus, they become different species. And so, this process of speciation, genetic isolation followed by genetic divergence, has occurred many, many times over the course of life on Earth. Until recently, we relied on fossil evidence to understand this evolutionary history, but nowadays we can use the DNA and the protein sequences of modern species to reconstruct these histories, and even look for signatures of recent selection and ongoing evolution. The ways DNA and protein sequences change over time, and how we use these changes to reconstruct these evolutionary relationships are our topics this week. But first, we need to review the way that we represent the evolutionary relationships between modern species. Phylogenetic trees are our first topic.